In this video, we're going to finish off the cell cycle by taking a look at some key differences between animal cell and plant cell mitosis, primarily focusing on cytokinesis. So we're essentially finishing cytokinesis, and we'll do a little bit of a summary to understand how to memorize the cell cycle in a better in detail, and how to understand the process in general, so it's not too much memorization and more understanding. So let's go back to cytokinesis for a minute, see the animals versus the plant cells. So in animals, that's where you actually see the pinching happen, as I mentioned in one of the previous slides in our previous video. You can see the pinching starting to happen there. The animal cells, they have a cell membrane, so that can actually pinch inwards. The physical manifestation of that pinching inwards is called a cleavage furrow. So you can see a little dip inside that's called a cleavage furrow. And there's a contractile ring. It's a ring of little muscle fibers that kind of pinches the cell into two. Again, the visual of that is that cleavage furrow that you see there. It's a little indentation that you see on both sides, and eventually the pinching becomes so pronounced and the cleavage furrow becomes so large that eventually that cell splits into two. So that's what you would see in animal cells during cytokinesis. During cytokinesis for plants, because there's a cell wall and there is still a cell membrane, there's still there, but the cell wall is very rigid, you're not going to see that pinching in. Instead, what you're going to see is, as I mentioned in the previous video, you're going to see a little cell wall start to form here. When the cell wall is starting to form, we call that a cell plate. And basically, the cell wall components come together in the middle, and eventually they join, and you have a new cell wall that separates the two cells. The cell plate, or the cell wall forming, comes from vesicles that come from the Golgi body of the cell. So again, there's another organelle helping out there. But again, you won't see the pinching in, you won't see the cleavage furrow, you'll just see a cell plate forming, which is one of the main differences between plant cell and animal cell cell cycle. There are more differences that you'd learn about at some other point, but right now these are the most important ones I want you to focus on. Again, here's a more visual of that under the microscope. You can see here the cleavage furrow happening because of the contractile rings pinching the cell into two during an animal cell. So you can see that here, becoming two, cleavage furrow. And here you can see a cell plate forming, and then eventually it all joins and connects to the other cell wall to make a new cell wall in between the two new cells. And so you can see the cell plate forming over here between the two new daughter cells about to be made. So remember that the example that we saw, we were looking at essentially 4 chromosomes. Keep in mind that we are organisms with 46 chromosomes in our cell. So our diploid number, 2n, is equal to 46. That if we had a cell going through mitosis, a cell cycle, our original cell would have 2n is equal to 46, and our two new cells that would form would also have 2n is equal to 46. So they'd also have 46 chromosomes still. And that's because what happens is during interphase, everything doubles. So at the end, when everything splits, you still have the same number of chromosomes. So, if we were drawing parts of the cell cycle with a human chromosome or a human cell, you'd have to draw 46 chromosomes. But we're not really drawing human ones. We're going to draw simplified cells with like 4 chromosomes in it, or 2, or maybe 6 sometimes. It's a lot simpler to draw than having to draw all 46 chromosomes in there. But the idea is that if you drew metaphase, you'd have to draw a metaphase with 46 chromosomes lined up in the middle, right? Because that's what would happen in a human cell. So as you can see here, this is all the chromosomes in humans, and then when they do make daughter cells, after mitosis, cytokinesis, you'd have also 46 chromosomes here and 46 chromosomes there. So this is our cell cycle summary. You can see this is a cell cycle here. We have interphase, then division. Interphase is that doubling part where the cell gets larger, doubles everything. You don't see much happening, but there's a lot happening there, 90% of the cell's life. Then you get into division. Division consists of mitosis and cytokinesis. Mitosis is really that prophase, metaphase, anaphase, telophase. and then cytokinesis is where that cell actually splits into two new cells, and it happens at the same time as telophase within this whole process of mitosis. So this chunk here is all this, and most of the chunk is basically the prophase, metaphase, anaphase, telophase, and then overlapping with telophase is that cytokinesis piece. And then these two new daughter cells, they could go through the cell cycle themselves and do the whole process again if that's what the body wanted. And then here you can see a comparison of plant cell cycle and animal cell cycle under the microscope. This is from an onion root tip because there's a lot of growth at the root area for the plant, so there's a lot of cell division. It's a nice area to get samples from. This is from a whitefish blastula growing. And so here you can see again interphase, nice defined nucleus, smooth. Interphase, nice defined nucleus and smooth. We go in the prophase. You can see more worms start to develop. It's not as defined as it was before the nucleus because it's starting to break down. Same thing here, not as defined, starting to break down, and you see more thick worms, which are basically your chromosomes. Go to metaphase, you can see the chromosomes nice and lined up in the middle. Chromosomes nice and lined up in the middle here. Go to anaphase, chromosomes are going to opposite sides, and you can see they're still chromosomes because they have the finger-like projections. Same thing here, very dark finger-like projections, but chromosomes are moving this way and that way. And then finally, telophase cytokinesis, you can see the chromosomes for telophase have become chromatin again, and you can see a nuclear membrane starting to reform. Same thing here, it's not as darkly stained as before, so we're becoming chromatin again. It's hard to see, but you'd see some pinching in happening here. You'd see a cleavage furrow for the animal cell. But over here, for the plant cell, you see a little cell plate starting to form in between the two, and so that's how you know it's a plant cell going through cytokinesis. So in this one page, everything was summarized with some cartoon images and some micrographs, so pictures from under the microscope. The only issue with this picture is that you don't see the spindle fibers and the centrioles, but keep in mind they are there. So again, here's some more practice that you can do. Study these pictures. Try to look at the pictures, look at the phase, and don't just memorize what phase goes with what picture, but also try to justify why each phase makes sense, right? Take a look at interphase versus prophase, right? If you look here, they look very similar, but here you don't see a, your smooth, defined nucleus like you had before. You see more dark stains now. Dark stains are over here, right? So you have dark stains here. The chromosomes are starting to sort of move apart, away from each other, whereas here you have your chromosomes lined up in the middle. Here you have your chromosomes moving to opposite sides. Here, it's hard to tell. Like, it still looks like you have chromosomes, so it's probably a bit of an early telophase happening. But you can see you have this this pinching in. So that means you must be in telophase because cytokinesis has started. So that pinching in is pretty pronounced here. And here you're basically, finishing up cytokinesis. So technically these two cells are probably in the same phase, just this is more advanced along here. So it's become more chromatin again. And then you have that membrane pinching even more because of that cleavage furrow happening here, which is important to understand. And again, same thing with the plant cell. Go through it and try to justify with clues why you think you're in the different phases, right? Try to cover up the names, cut them up if you want to, mix them up, and then try to put them in the proper order. And that's really good practice for trying to see the difference between several phases. And then a few tricks for you to remember the order of things. So you can use IP, MAT, and then C. IP, I is interphase. MAT would be the mitosis part, which is prophase, metaphase, anaphase, telophase, and then C is that cytokinesis part. Some students like to use the I, prefer mice and talking cats. So I is interphase, prefer prophase, mice metaphase, and anaphase, talking telophase, and cats cytokinesis, but IP, MAT. And then remember to add the C there for cytokinesis. So again, all of the components are summarized in this one slide here. Go over it, try to remember this trick, and it's important for you to actually try to draw out the phases and label things. That'll help you to memorize and understand rather than simply memorize and not understand. You can watch this video here to recap the process. It's a separate video that was made. There's lots of videos out there that go over this with some nice clear animations, so if you need other support, please make sure you do that. Some recommendations for your lab notebook when you're doing your summary, depending on what the direction was from your teacher, but oftentimes you'll be asked to do a 2-page summary for the entire unit. When you can draw out the cell cycle, the actual chart, label the main parts, so interphase versus the division part. Can you describe that chart? Can you describe the main phases during the main differences between plant and animal cell cycle, especially at the level of cytokinesis? Can you draw this chart and then around it draw the different phases? Because that way you can consolidate it and summarize it in a way where everything is together as opposed to separate. So you understand how everything is connected together. How does this picture relate to this picture? How do these pictures relate to all the other pictures we saw in this slide deck? And if you think about it, this part here represents essentially the prophase, metaphase, anaphase, and a little bit of telophase. And then this part right here going to there represents that cytokinesis piece. And this to this represents this whole interphase piece. So try to see how they're all connected to each other.